I'd like to welcome you here to the Environmental Science Lab on the North Metro campus. Come on in and we'll take a look at some of the things that you're going to be dealing with in this program. So one of the first things you're going to get hands-on with in this program are going to be microscopes. You're going to have to be able to look at samples in a microscope and identify bacterial counts or microorganisms such as amoebas, filamentous bacteria, so on and so forth. And then also over here, something we talk about a lot of times are our handheld devices that we use probes where we measure stuff like pH, chloride, nitrate, nitrite, your LDO, so on and so forth. These are used both in the field and in a lab setting. So next, we'll look at something you'll become very familiar with in this program, and those are our incubators. Almost everything that we do biological has to be incubated at some point in time. If you look inside here, we have some of our ColiSure samples to test for fecal. These cook for 24 hours, and then we will analyze these under the black light box that I'll show you in a little bit. One of the things you have to do in all of our treatment programs here is analyze E. coli. So you would take these and put them in the black light box and you would analyze how many of these wells are glowing, and then there's an actual sheet to convert this number to an MPN of E. coli. And if you come on over, you can look down in here and see what I'm talking about with the glowing. Two of the next things we look at are used heavily in the water treatment industry. This is called a jar tester. This is what you use in a lab to determine how much alum or coagulant to dose your water with in treatment. And this over here is our water bath, where we can heat waters up to the temperatures required for individual tests. The next step in our journey is one of the first things you would have done before you hit the incubator with those samples that I showed you, and that's actually make your sample. You would collect your water, you would mix in the reagent, and then you would put all of that into this tray and then send it through the tray sealer before you take it to the incubator. Next, we'll just look at some of the items that you're going to use out in the field when we're not in water, and just laid out some of them here so that you can get a sample of it. A lot of this relates to trees because that's one of the most common things we do here when we go out into the wild is take a forest mensuration, which is basically determine what type of forest it is and how healthy the forest is. Some of the things that we'll have to use, we use this item here to figure out the canopy cover in a forest, calipers to get the diameter of a tree. You have to be able to orient yourself with something other than a Google Maps feature. You have to do it with paper maps because technology doesn't always work. Signals don't always reach where you're going to be going. So I'm going to teach you map reading, going to teach you topography, how to orient, how to use a compass, how to navigate yourself in the field so you can get from point A to point B and end up where they want you to be. Remember, this program is water-centric, so a large portion of our curriculum always going to go back to water. And this is just a small sample of some of the items you could possibly use while you're in this program. This here measures the flow of water. You insert this into a column of water. The propeller will spin. You will get a number of rotations up here on the measuring device. This is called a staff gauge. It allows you to get a quick, easy measure of the column of water. We can use this out in the field for smaller streams that don't have permanent staff gauges. So most students' favorite piece of equipment when we get out in the field is this backpack. It is an electrofishing backpack. This is what we use to sample the fish in our aquatic environment. It'll have a battery in it. We go out into the field, we analyze the water, we determine what voltage we need, and then we use the wand. When you depress the button, it sends an electric current through the water that will stun the fish. When you let off the button, it immediately stops the electricity going to the water. We will grab the fish in our buckets and then take them to a spot later to analyze those fish. While you're doing this activity of electrofishing, you will have to wear waders to protect you from the electric current. The college provides waders, but you're also more than welcome to bring your own if you have a personal set. Thank you for taking the time to view some of the items we'll do here in the Environmental Science Lab. I look forward to seeing you in this lab or in the field. If you've got any questions, please reach out to me.